let's just start.
Okay. You want to do it?
Yeah, sure.
Go ahead.
1, 2, 3, start recording. Did you
I,
start?
I already started.
What the chaos, Okay. Hello everyone. Welcome to the we don't have a name.
We do. It's midterms in a pod.
Is it confirmed? Is it confirmed?
Yeah,
Okay,
yes,
okay.
it's confirmed.
Welcome
Midterms.
to the Bio 1 episode of Midterms in a Pod, the podcast, which is a collab between Steady Sheep and Addendum Marianopolis. So let's straight up get into it.
Okay, wait. Let me just put out a disclaimer. We are students. Yeah, that was weird
Yes.
grammar. Anyway, let's start.
No, that was great. Also, first of all, maybe a quick introduction on who we are. So, I am Camille. I am a first year in Arts and Sciences. And what about you?
Oh, okay. I'm her friend. My name is Melissa. I'm also in first year, and I'm also in arts and sciences. And it took us like 1 minute for us to introduce ourselves. Wow, this is great time management.
Yeah, second disclaimer. So, basically, we took Bio 1 in our first semester. So, there might be like some little inconsistencies like maybe towards the ending. Maybe there's stuff that you haven't viewed. So don't panic about that if we do cover stuff that is extra. And yeah, let's get into it.
Jesus, that took so long. Okay, carbohydrates. So basically, carbohydrates are macromolecules, obviously, and there are two types. There are monomers and polymers. So Cam, what is a monomer?
Monomers are like a molecule composed of one unit with like a hydrogen end and a hydroxyl end.
So then we have a polymer, which is made up of a lot of monomers, and they're basically, very large molecules, okay? And they also have a hydroxyl end and a hydrogen end. And there are two types of reaction that happens with monomers and polymers. There is a dehydration and hydrolysis. So what is a dehydration?
Dehydration is when we drain monomers together to form a polymer. So they go like poof 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 and poof.
Yeah.
Yes.
So yeah. And hydrolysis reactions, what is that?
It's when we break down polymers to release monomers. So they once again go poof poof poof, but it's the reverse reaction of dehydration.
Yeah.
All right, so that was macromolecules. Let's actually get into carbohydrates.
Mm-hmm.
So, what are the monomers in the carbohydrates? Like, what are they called?
So specifically in carbohydrates, they are called monosaccharides. Yeah, that's a long ass word. And the polymers are called polysaccharides. And an example of a monosaccharide, so a carbohydrate monomer, would be glucose. So there's also disaccharides, which is basically 2 units. It's a polysaccharide technically, but it's only 2 units.
And examples of disaccharides are usually sucrose, which is formed from glucose, and fructose, which are both monosaccharides. And then lactose, which is formed from glucose and galactose. Let's get into actually the different types of polysaccharides. So we have 2 types of polysaccharides that both start with S. What are they?
Oh my god. Oh, this is so far into my mind. A type of polysaccharides, okay, is there's one that's a storage polysaccharide and one that's structural polysaccharide. So storage for storing energy and structural for building material,
Yes, sir. And then in storage polysaccharides, the 2 main polysaccharides that we'll be seeing usually in class is starch and glycogen. And for structural, it's usually cellulose that we cannot digest, and chitin, which forms like amino acids and amino sugars and such. The second type of macromolecules is lipids, which are composed of 3 different types.
Okay, so the 3 different types are fats and oils, phospholipids, and steroids. Basically, it's composed of glycerol and 3 fatty acid chains.
Drained by 3 ester linkages, but they're formed by dehydration. So you have 2 types of fatty acids. Basically, you have saturated fatty acid and unsaturated fatty acid. Saturated equal bad and unsaturated equal good. All right.
Right.
Moving on to phospholipids. So phospholipids are
Amphipathic. So amphipathic means that there are two behaviors, which is true because the phospholipids are like they have a head and like two little tails, and the head is hydrophilic, which is water loving, while the two little tails like that look like legs are like two fatty acid chains, which are hydrophobic, and they make up the phospholipid bilayer, which is the major component of a membrane.
So steroids aren't, something very special about steroids is that they're not a monomer nor a polymer. Yeah, that's all you need to know, honestly.
Yeah, honestly, that's literally it. Some type of steroids that are being made, well, animals make cholesterol, which are used to make other steroids like sex hormones, so testosterone, estrogen.
Alright. Proteins are responsible for the activity in the cell. So amino acids are the monomers, basically, of proteins, and
a central carbon attached to an amino group, a carboxyl group, hydrogen, and a side chain that varies to make the different 20 amino acid, which is called the R.
Yep, and they're linked by what's called a peptide bond.
Yep.
So then, onto that, we have polypeptides, which are like the polymers of proteins.
There are 8 functions, though, of proteins. Want to go over them real quick?
Okay, so very quickly, the 8 functions of proteins are structural protein, storage, transport, hormones, receptor, antibody, enzyme.
You guys might have one over like the thing, like there's a key or a substrate key or whatever. It's not that important. We never had any questions on it.
And movement.
Yep. Next, nucleic acids. Wait, are we doing that?
Yeah, nucleic acids. So once again, in nucleic acids, you also have monomers and polymers. The monomers are called
nucleotides.
Alright, so let's go over the nucleotides first. The nucleotides are composed of 3 different parts, and that's super important.
Yep. So there are the nitrogenous base, which is composed of 2 types again, which is the purines, which is A and G, and the pyrimidase, that word I do not know how to pronounce, which is the rest, which includes C, U, and T. It's important to know that U only appears in RNA, while T only appears to DNA. So, Cam, can you tell me which one binds with which? Like, which letter binds with which letter? Just a refresher.
Yes. A binds with T, and U, if it's in RNA, and G binds with C.
Yep.
So,
a second part of nucleotides are the sugars. And lastly, there's a phosphate group. That's important, and that's part of it as well.
Okay, so DNA and RNA are the polymers of nucleic acids. So basically, monomers are nucleotides, and then polymers are DNA and RNA. So that's, yeah. You need to quite know, like, the structure of DNA and RNA, because usually there's, like, quite a bunch of questions on that. Do you want to go over the structure of DNA?
Oh, okay. So basically, the DNA structure is it's like 2 double helixes. So 2 strands of nucleotides. So there are 2 sugar phosphate backbones, and nitrogenous bases are in the center. And like we said before, A binds with T in a DNA, and G binds with C. So yeah. Some functions of DNA we all know already is inheriting the genetic information, controlling the activity of the cell. The genes are located in the chromosomes, and then it also contains the recipes. Now let's move on to RNA.
Yeah, so RNA are like one single strand of nucleotides instead of 2, like DNA. And they, for their like nitrogenous base, they have U instead of T, like Mel said earlier. And their function is mainly protein synthesis, so like producing protein, which is very important. And yeah, I think that's basically it. Oh, you have 3 types of RNAs. You have the mRNA, which is the messenger RNA, the tRNA, which is the transfer tRNA, and the rRNA, which is the ribosomal RNA, which is the site of all protein synthesis. So that's super important. So yeah, just know that. And let's do transcription. So transcription is when DNA becomes an RNA.
So basically, like she said, it's when DNA becomes RNA. I'm kind of repeating that, but that's really important. And how they do is they copy one side of the DNA.
And it happens in the nucleus, so that's usually pretty important. So there's this translation. Translation is protein synthesis. Basically, it's when we change the sequence of nucleotides on the mRNA to make a polypeptide.
Yeah, that's basically it. Okay, next. ATP. ATP. So basically, ATP is the cellular energy that's needed to do cellular work. And yeah.
Yes.
That's it. That's all you have to know, honestly. Are we now onto cells?
Yeah, let's get into cells. It hasn't.
I
it's not
hate
like
this
we're
part.
recording
I hate.
30 minutes. Wait, you hate cells?
I hate cells so much. Okay, that is not important. Okay, so there are two types of cells. There are prokaryotic cells and eukaryotic cells. And prokaryotes are organisms composed of prokaryotic cells, like obviously, and they're found in two domains: bacteria and what the heck? Archaea. So important. Prokaryotic cells do not have a nucleus and they do not have membrane-bound organelles, okay? That's the two most important traits of it. They do have a DNA, and they have ribosomes, and they have a plasma membrane, and they also have a cell wall composed of peptidoglycan. So, yeah.
Yep. So that's usually pretty important. There's always a question on that. Okay, eukaryotes. Eukaryotes are organisms composed of eukaryotic cells, and they belong in the domain eukarya. So if we were to compare an animal cell and a plant cell, which both are eukaryotes, by the way, the main difference would be that an animal cell has the two C's.
The two C's, which are centrosome and centrioles.
Yeah, which plant cells don't have. And on plant cells, they have like the MOC, the NOC, a cell wall, and a chloroplast.
Oh, and also their vacuoles are different, I think, in each one. But they have a lot more in common, and we're going to go over them in a bit. So basically, yeah. Also, important difference between eukaryotic and prokaryotic is that eukaryotic cells have a nucleus, and they have membrane-bound organelles, unlike the prokaryotic.
Yes, so now we get in the very extensive list of eukaryotic organelles and structures. We are starting with the center of everything, the nucleus, which, apart from containing all the important stuff like the DNA, and controlling the activity of the cell, is basically it.
Site of transcription too. Nucleolus, which is also is it within the nucleus?
Yeah, it's in the nucleus. Which is kind of our RNA synthesis. Yeah, let's get into ribosomes, which are, you have two types of ribosomes, free ribosomes and bound ribosomes, and all protein synthesis begins on a free ribosome. So they become bound ribosomes.
So there's the RER and the SER. You don't really have to know what each letters mean, just know that one it means rough and the other is smooth. And basically for the RER, so the rough ER, it's protein exportation. It's where there's a synthesis of glycoproteins, while the smooth ER is the synthesis of lipids. And yeah, and they also have a detoxification function.
Let's get into the Golgi or Golgi. I don't know, people say it differently, but the main thing to remember is that it basically doesn't do anything. Yeah, you don't really need to know it. You just need to know that it's kind of like an intermediate between like the RER/SER and the lysosome.
Then there's the lysosome, which is part of the Golgi apparatus. What they do is they recycle broken organelles back to monomers, and they're also programmed for the destruction of cells. And they do phagocytosis, they do cellular aging.
Vacuoles in animal cells, you have two types. You have food vacuole, which is where phagocytosis happens because that's food, that's eating, and contractile vacuoles, which are contractile vacuoles. We won't discuss that further.
The vacuoles in plants are called large vacuoles, and they have storage abilities, and that's where it's a disposal site basically. Okay, yes. Endomembrane system is important. Like they're always going to ask you questions about what's part of the endomembrane system, and it's very important to know that it's nucleus, the nucleus, RER, SER, the vesicles, the Golgi, lysosomes, and vacuoles are part of the endomembrane system. Okay, that's just very, very important. It's like basically the transportation system, you know?
Yeah, and you need to know the order because they always put that little drawing with the order. You we are now getting into the
Peroxisome?
Peroxisome? Peroxisome? I like it. Yeah, that word. You guys know what I'm talking about. So, what does it do? It doesn't do anything important. It just breaks down alcohol and lipids. Okay, not important. Moving on. The chloroplast, which is only in plants, that is that is where photosynthesis happens.
Mm-hmm.
Yeah.
It's surrounded by two membranes. The chlorophyll are in thylakoids and thylakoids. Thylakoids had some questions on those, but it's like little details like that that are not too hard, especially if it's an open book exam. It's not too hard to find. Oh yeah, no, we're something about chlorophylls. They have two types of reaction. They have the light reaction, and they also have the dark reaction. So I don't know if you guys went over it, but there is a drawing of what a chlorophyll looks like. So it's not that important, but it'd be interesting to understand it.
Okay, the mitochondrion or mitochondria, depending on plural, singular. Cellular respiration.
Cytoskeleton, it's like a network of protein fibers, and there are three types. There's the microtubules, microfilaments, and intermediate filaments. And they have functions, important functions, okay? They have the bone function. They also have the muscle function.
Let's get into the centrosome, which, if you remember, is only in animal cells. So the centrosome makes the cytoskeleton. And it also makes a mitotic spindle during cell division, which we're going to see later. And then we have the MOC, the MOC, which is only in plant cells, and it's basically the centrosome, but in plants.
Mm-hmm. Centrioles are part of the animal cells only, and basically it helps with the assembly of the mitotic spindles. So yeah, it's there. The plasma membrane and transport. So there's the fluid mosaic model to describe the plasma membrane, basically, which is like a collage of proteins, and it's mostly made of phospholipid bilayer. Okay, that's important. So there are integral in the membrane. There are integral proteins, which is for the transport of in and out of the cell. So there are channel proteins, graded channels, and aquapoins for water permeability. And then there are also carrier proteins for passive transportation, called carrier facilitative diffusion. And the carrier proteins are also used for active transport. And then there are peripheral proteins, which is not for transport, and it's like a receptor.
So those are the four main things I think that you need to know are in the phospholipid bilayer. So they always put an image of the phospholipid bilayer with these four elements. So cholesterol, integral protein, peripheral protein, and glycoproteins or glycolipids. And you have to either name them or say what they do. So know that.
Passive transport and active transport. Passive transport is when we go from a high concentration to a low concentration, while active is the other way around, so from low to high. And basically passive transport is passive because there's no ATP required, while active transport is ATP is needed because you're going against the concentration gradient.
So you have simple diffusion, which is small, usually smaller fat soluble molecules, and you just go through the phospholipid layer, they just through it. And then you have carrier facilitated diffusion. They go through a specific integral protein.
Mm-hmm.
And finally we have osmosis, which is the movement of water through a semi-permeable membrane. So it's when you go from high to low concentration.
So basically osmosis, they bring about hypotonic, isotonic, or hypertonic solutions. And what happens for plant cells in a hypotonic solutions, which means it's a lot of water, it's really good for the plant cell. They're turgid, they're alive. If they're in a isotonic solution, which is just there's a bit of salt in there, it turns flaccid, which is not good for the plant cell. And finally, in a hypertonic solution, which there's an increase of salt, it dies. As for the animal cells in a hypotonic solution, which means there's a lot of water, when there's a lot of water, it dies, it bursts, and that's called a hemolysis death. While in an isotonic solution, it's healthy, and finally in a hypotonic solution, it shrivels up. So it doesn't die, but it's not good for it either.
You recommend just having a drawing and basically being able to
Yeah.
see it. Like visual is, yeah.
There will
Alright.
be a number on that, I guarantee.
There's always a number on that. Okay. Active transport. So active transport, you have exocytosis and endocytosis.
Endocytosis, which has like two types, which includes the phagocytosis and the pinocytosis. Okay, cell cycle and mitosis. Okay.
Cell cycle is like basically your lifetime. It's divided into usually two main phases: interphase and mitosis. Interphase is like 99% of their lives, and mitosis is their like reproduction.
Right, interphase is like in between cell division. Basically, there's a G1 phase. That's when the daughter cell is growing. So there's transcription and translation. Then there's the S phase, which is the DNA replication. So both sides of the DNA are copied. And finally, there is the G2 phase, which is basically cellular growth. They double the organelle, so each daughter cells has a copy of each other. DNA is uncoiled and called chromatin. I remember a question like that. Mitosis! Let's go. It happens in, well, animals, obviously, and it forms all the cells of the body, so the somatic cells, except the gametes, where we're going to talk about meiosis afterwards. And yeah, it's basically diploid cells. 2 sets of chromosomes, 1 set from the mother, 1 set from the father. Those are little details, honestly. And they always produce identical daughter cells, so that is important.
And so mitosis is composed of 4 main phases, that which is prophase, metaphase, anaphase, and telophase. So do you want to kind of like quickly go over?
Okay, so prophase is the phase where like we see the chromosomes. We see like that they each contain 2 sister chromatids, which is like the 2 identical DNA molecules attached, and they're just kind of there, and they're biocentromere, where the chromatids are joined together, basically.
Metaphase is when like the individual chromosomes they align and at the metaphase plate, which is like the center of the cell, and they align in like a single file. So or Sumi always used to say "I like your daughter," so that's what happened.
And then there's the anaphase, which is where the sister chromatids separate from each other and move to the opposite poles, and each chromatid is now considered a single chromosome. So technically, the number of chromosomes should be doubling.
And finally, you have telophase, which is where cytoplasm division happens. So the cell actually like divides into 2. In animal cells, it's called like a cleavage for
Yes. Yes.
yes. And it ends up forming 2 identical daughter cells.
It's a lot of information. Highly recommend to just have like a little drawing next to you to see like what's happening in each one of them.
Yep.
Because there's always gonna be questions about it, especially on like.
Okay, so meiosis is like when it's basically when the gametes form.
Yes.
So it always starts with a diploid cell, so like a normal cell, and it ends up undergoing 2 nuclear divisions, so meiosis 1 and meiosis 2, to basically produce 4 genetically different haploid cells.
You just really have to know what meiosis 1 is, because that's a reducing division, while meiosis 2 is basically the same thing as mitosis.
Exactly. So meiosis 1, 4 phases: prophase 1, metaphase 1, anaphase 1, and telophase 1.
So basically, in meiosis, the difference in prophase 1 is that there's a synapsis going on, which is basically the joining of the homologous chromosomes. So they're going to be forming tetrads, okay? And then the homologous chromosomes have the same genes in the same sequence. Then there's also the crossing over, which is when the homologous chromosome exchange genes. And that's like the first source of genetic variation. And it's like, it's like, what's the word? Au hasard, okay? It's au hasard. I don't know.
It's random.
It's random. There you go. It's randomly done.
Yeah, so basically prophase 1, the main thing to remember is that synapses and then crossing over, which is always super important. They always talk about when does genetic variation happen and stuff. So yeah.
Then there's metaphase 1, which is basically when tetrads align at this metaphase plate, and that's when there's the independent assortment, which is basically members of tetrads align randomly, and that's a second source of genetic variation. So yeah. And then there's anaphase 1, which is where the tetras separate and move to opposite poles. there's telophase 1, which is the cytokinesis. So there's a cytoplasm division forming two genetically different haploid daughter cells that will undergo meiosis too.
Yes! Alright, so let's get into meiosis 2, which is basically the same thing as mitosis. Phase 2, metaphase 2, anaphase 2, and telophase 2, and it's really the same thing as mitosis. I don't think there's much difference. No, there isn't any difference.
Just
So,
at the end, the only difference that they yield 4 genetically different daughter cells.
yeah,
Just-
that's important.
Yeah, that's important.
That's the main difference. Yeah, okay, so let's just stop here. This was a mess, but it was fun nonetheless.
That was so bad. Keep in mind that
we're putting that in. We're keeping that. That was such a good outro. Okay, no, for real though. Thank you guys if you've stuck until now. Hopefully there's someone who's still listening to us. Yeah, okay.
Wait, no, but I'm losing my mind because like this is so long. This is gonna be so long to edit. Oh my god.
So messy. Yeah. Okay. So basically, thank you for everyone who stuck here, and hopefully this helped. We are very new at this, and we're just surviving on our last brain cells, so it's a bit
It's a bit
But,
everywhere. It should
yeah,
be entertaining.
but we tried our best. We hope that you liked it, or that it helped, or that it made you fall asleep. Like if it made you fall asleep, good for you too. That's it. I think that's it. Thank you for tuning in, and stay tuned for more
Capsules.
exciting stuff from
Right.
Study Sheet and Addendum. Just gotta promote the clubs. Okay,
Oh.
that's it. Bye.
Thank you for listening. We really tried our best, okay? So you take all of the information that we tell you, not with a grain of salt, but like, just accept it. Anyway, I wanna cry.